Hello everyone and welcome to Roadmap. So today's topic is secondary metabolites part 2, and if you like the video, don't forget to click on thumbs up button below. You can also subscribe for more video updates. Topics to be covered are production of secondary metabolites, selection of cell lines for high yield of secondary metabolites, visual or chemical analysis, selection for resistance. Production of secondary metabolites. Now, the process of in vitro culture of cells for the large-scale production of secondary metabolites is complex and involves some of the aspects, such as the selection of cell lines for high yield of secondary metabolites, large-scale cultivation of plant cells, medium composition and effect of nutrients, elicitor-induced production of secondary metabolites, effect of environmental factors, biotransformation using plant cell cultures, and secondary metabolite release and analysis. Now, selection of cell line for high yield of secondary metabolites. The very purpose of tissue culture is to produce high amounts of secondary metabolites. Now, majority of callus and suspension culture produce less quantity of secondary metabolites, and it is mainly due to the lack of fully differentiated cells in the cultures. Now, some special techniques have been devised to select cell lines that can produce higher amounts of desired metabolites. Now, these methods are ultimately useful for the separation of producer cells from the non-producer cells. The techniques commonly employed for cell line selection are cell cloning, visual or chemical analysis, and selection for resistance. Now, let's look at it one by one. Coming to the cell cloning. Now, this is a simple procedure and it involves the growth of single cells taken from the suspension cultures in a suitable medium. Each cell population is then screened for the secondary metabolite formation, and only those cells with high yielding ability are selected and maintained by subcloning. So, coming to the single cell cloning, there are certain practical difficulties in isolation and culture of single cell aggregate cloning. Compared to single cell cloning, cell aggregate cloning is much easier and it is preferred by many of the workers. Now, coming to the visual or the chemical analysis. A direct measurement of some of the secondary metabolites produced by cell lines can be done either by visual or chemical analysis. Now, the visual identification of cell lines producing colored secondary metabolites, pigments, example the beta-carotene and chicinin, will help in the selection of high-yielding cells. This method is quite simple and it is also non-destructive. And the major limitation is that the desired metabolites should be colored. And there are certain secondary metabolites which emit fluorescence under UV light, and the corresponding clones can be identified. Now, some workers use simple, sensitive, and inexpensive chemical analytical methods for quantitative estimation of desired metabolites. Now, analysis is carried out in some colonies derived from single cell cultures. Radioimmunoassay is the most commonly used analytical method, and the microspectrophotometry and fluorescent antibody techniques are also in use. So this is about the visual or chemical analysis. Coming to the selection for resistance. Now, certain cells resistant to toxic compounds may lead to the formation of mutant cells, which can overproduce a primary metabolite and then a secondary metabolite. Such mutants can be selected and used to produce the desired metabolite in large quantities. So, one example such as the cell line selected for resistance of 5-methyltryptophan, which produces strains which can overproduce tryptophan. Now, these tryptophan overproducing strains can synthesize 10 to 50 times higher levels of the natural auxine, namely indole acetic acid. So, this is about the selection for resistance. Now, this is a schematic representation of cell aggregate cloning for the selection of high-yielding cell lines. Now, what happens exactly over here is a high-yielding plant of the desired metabolite is selected and its explant are first cultured on a solid medium. After establishing the callus culture, high metabolite producing calluses are identified and they are grown in suspension culture. Cell aggregates from these cultures are grown on solid medium further, and then the freshly developed cell aggregates, that is the calyces, are divided into two parts. One half is grown further while the other half is used for the quantitative analysis of the desired metabolite produced. And the cell lines with high yield of secondary metabolites are selected and used for the scale-up and suspension culture. And then it is further followed by large-scale tissue culture in a bioreactor. So this is about the schematic representation for the selection of high yielding of cell lines. And I hope that you are enjoying the video. Stay tuned for more lessons to come. If you have any doubts, feel free to ask in the comment section below. Thank you for watching.